So I'm going to talk about the HER2 DNA breast cancer vaccine. It was discovered by the CVI team, which is the University of Washington's Cancer Vaccine Institute, and also by a guy named Dylan. So I'm going to start off by saying that cancer patients and researchers alike have been dreaming of the day when treatments can be developed that are not only effective but are also affordable with low toxicity luxuries that haven't typically accompanied effective cancer therapeutics treatments in the past. But the recent work from the University of Washington's Cancer Vaccine Institute, CVI, which is part of the Cancer Consortium Breast and Ovary Cancers Program, show that the day might not be as far off as it seems. So they did recent studies in their oncology studies, and they showed promising results from a non-randomized clinical trial for a breast cancer vaccine in advanced stage of HER2 positive breast cancer patients, and they redid a 10-year follow-up. So they proved that the vaccine can be effective, but only to a subgroup of people with breast cancer. And although limited to a small number of patients, the results from the trials are somewhat promising. So what's interesting about this study is that before they were able to develop the promising HER2 DNA vaccine, uh, figuring out how to make this vaccine against cancer took some trial and error. Dylan, the guy that I talked about earlier, he explained that the real breakthrough came from understanding of how the same protein can provoke different immune responses. For instance, one part of the protein may elicit a strong tissue destructive response that kills off cancer cells, while another part of the same protein may elicit and promote disadvantaged immune suppression. But Dylan said with the CVI team that he figured out this system, and now he got a hammer to combat cancer. Those are his words. So this is for the introduction. I'm going to start off with some definitions that are going to help understand better concepts later when I'm going to talk about the tests and the interpretations that will run up by the vaccine. So what is triple negative breast cancer? It's when in the breast we have no presence of estrogen and progesterone hormones, and also the breasts are negative for human epidermal growth factor receptor 2. What is the DFS rate in ITT? So DFS is disease-free survival rate in the intention to treat. The intention to treat is just the difference between the treatment group, so the vaccinated group, and the control group mean. So we're going to also run some tests for IHC 3 plus FISH plus patients. What is these patients? So what does that mean? It's immunohistochemistry test, which proves it gives a score from 0 to 3 plus, and these numbers, they measure the amount of HER2 receptor protein on the surface of cells in a breast cancer tissue. For example, so if it's 3 plus, then the patients have a lot of HER2 receptor protein. And if the patients have a positive FISH test, which is the case here, then he has the breast cancer cells make too much of the HER2 protein responsible for the breast cancer. So now the HER2 DNA vaccine, what does it contain? The GM-CSF, which is the granulocyte macrophage colony stimulating factor. What does it do? What is it? It's a cytokine that promotes myeloid cell development and maturation, which is gonna help the immune system later. And the HER2 DNA vaccine also contains the AE37. It's a HER2-derived peptide based on another hybrid peptide, which is AE36. So what does this peptide do? It enhances antigen-specific stimulation, so it helps the immune system, and also enhances a CD4 TH response. So these cells are helper cells. They help with the white blood cells and all of that. The GP2 is also present in the vaccine. It's also a derived peptide of the HER2 DNA. But this time, the GP2 elicits a CD8 plus response. The CD8 plus are cells that help kill the disease cells, the cells that have been caused by the disease. So a little bit of like, we're going to talk about the timeline of how this vaccine was discovered. So in 1887, there was the first identification of the HER2 clinical gene of breast cancer. Then in 2009, they discovered the utility of the peptide that I talked about earlier, which enhanced TH responses. In 2016, they run their phase 2 trials of the peptide E37 that prevents recurrence. And in 2020, the phase 2 trial of 2 HER2 peptide vaccines were in place, the GP2 and the AE37, which has proven that they prevent recurrence, so they help with the immune system memory. So the primary goals that were searched by the CVI team and Dylan were that the vaccine activates the immune system, of course, it prevents recurrence, and also it increased the disease-free rate of HER2 patients. So with the tests and experiments, they enrolled a total of 298 people. Some were the control group, which received only the GM-CSF, but others received the vaccine, which is the peptide AE37 and the GM-CSF. For the first test, they searched for the level of toxicity difference between the control group and the vaccinated group. So it's a little bit flu now, but if we see in the bigger picture, the vaccinated group shows a decrease in toxicity compared to the controlled group in green. So what does local toxicity and systemic toxicity means? The local means that it affects neurovascular manifestations, paresthesias, and anesthesias. The systemic toxicity most affects the nervous system or the cardiovascular system. They also did some tests concerning the immunologic response to the vaccination. So they saw that with the vaccinated people, the vaccinated patients, an important increase in the delayed type hypersensitivity DTH reaction. What does DTH mean? Delayed type hypersensitivity is defined as it's the recruitment of T cells that are gonna go into tissues to be activated by antigen-presenting cells to produce cytokines. So basically, the CD8 plus T cells are now present to help more and more with the immune response and with the defense against cancer cells. Furthermore, if you can see in the B figure, how the T-cell proliferation, and with the BC figure, of course, also, how the T-cell proliferation response is much more important with the vaccinated group than with the control group only. So we can clearly say that the AE37 peptide and the vaccine, of course, provokes T-cell proliferation and the DTH response. Additional immunological responses. Now we're going to focus on the IFN gamma cells. So we can clearly see in figure A how the vaccinated group shows a more important concentration of IFN gamma cells than with the control group only. And we also can see in the figure number B, named B, in the third graph, how the presence of TRAC cells is more important in the vaccine, in the control group than in the vaccine group. So we can say here as an interpretation that the AE37 derived peptide enhance IFN gamma cells, which will activate macrophages. This is the role of IFN gamma cells. And these macrophages are gonna enhance the phagocytosis of cells with the disease, cancer cells, antigens. And the AE37 derived peptide also provoke a decrease in TRAC cells, which provoke a decrease in immunosuppressivity and enhance the autoimmunity of the body of cancer patient cells. The last experiment that they did concerned the disease-free survival time. Now, we can see that for figure A, for all patients, for patients with HER2 overexpressing breast cancers, for patients with HER2 1+ and 2+ tumors, there wasn't a significant differences between the vaccinated group and the control group. However, in figure D, we can see a significant response with the triple-negative breast cancer patients. So we can see that with the vaccinated group, the disease-free survival rate is much more important than with the control group only. Here are some pictures that I found. The breast cancer patients that are enrolled in CVI trials at various patient advocacy and reunion events. I can say as a summary that the overall intention to treat analysis, so the comparison between the control group and the vaccinated group, demonstrates no benefit to vaccination that is important. However, the results confirm that the vaccine is safe and suggest that vaccination may have clinical benefits in patients, but only with a specific subgroup, which is the TNBC, the triple-negative breast cancer patients. So this phase of trial reveals that the AE37 vaccine is safe and is possibly associated with improved clinical outcomes of disease-free rate in certain subgroups of breast cancer patients. Now, we cannot forget that every journey starts somewhere, and this can be a start of something. It's not important now as the results show, but it showed a little bit of hope with the triple-negative breast cancer patients. Thank you.